Hello bachcho, let's see the question. So the question says, which one of the following has found extensive use in genetic engineering work in plants? So इन चारों ऑप्शंस में से हमें आइडेंटिफाई करना है जो प्लांट्स के जेनेटिक इंजीनियरिंग में यूज किया जाता है. So the key concept in this question is to firstly understand the cloning vectors which are used for plants. So plants में जो सबसे commonly used cloning vector है वो है Agrobacterium tumefaciens. So this is a type of bacterial species which causes a very specific type of disease in plants, which is the crown gall disease. So in crown gall disease, there is formation of tumor in the plant cells. So the genetic factor which is responsible for causing this tumor disease, that is the crown gall disease by Agrobacterium tumefaciens, is the presence of a plasmid known as Ti plasmid in the bacteria. Now, Ti plasmid is used as the most common cloning vector for plants. Now, this is because the natural capability of Ti plasmid to be transferred from the bacteria to the plant cells. So, while the Agrobacterium tumefaciens infects the plants, it results in the transfer of the Ti plasmid into the plant host cell genome. So in this way, we can exploit the Ti plasmid for our own interests. So if we look into a little bit more detail and the Ti plasmid structure, then we will better understand how Ti plasmid is utilized in genetic engineering. So the Ti plasmid has two important regions. So the पहला वाला जो सबसे important region है Ti plasmid में वो है tDNA or the transfer DNA. ना इसे ट्रांसफर डीएनए इसीलिए बोला जाता है because this is the part of the Ti plasmid which can be transferred from the bacteria into the plant cells. And the second region which is present in the Ti plasmid is known as the Vir region or the virulence region. So the Vir region is responsible for the process of transformation. So when the agrobacterium infects the plant cells, this virulence region is responsible for the excision of the tDNA from the Ti plasmid, and then this tDNA is transferred into the host cell genome, that is the plant cell genome, where it gets incorporated into the plant cell and then causes the crown gall disease in the plant cell by formation of the tumor. So the tDNA of the Ti plasmid is responsible for causing the tumor. So the tumor genes are present in the transferred DNA region of the Ti plasmid. Now, how can we use this Ti plasmid for genetic engineering? So what we can do is we can replace the tumor genes present in the tDNA with our gene of interest. So if we do that, then what will happen is that the virulence region will work the way it does. That is, it will cause the excision of the tDNA, and then this tDNA with the gene of our interest will be transferred into the host cell genome, that is the plant cells. So therefore, the TI plasmid is like a cloning vector. So just by replacing the tumor genes in the transferred DNA region with the gene of our interest, we get a recombinant TI plasmid, and this recombinant TI plasmid is utilized in transferring the gene of our interest into the plant cells, and therefore it helps in genetic engineering or the recombinant DNA technology. So the recombinant TI plasmid is acting like a cloning vector. So we can say that the species which is most commonly used for genetic engineering in plant cells is Agrobacterium tumefaciens. So the first option, that is Bacillus coagulens, is not utilized in genetic engineering for plants, so this won't be our answer. The second one, that is Agrobacterium tumefaciens, is the one that is used for the genetic engineering in plants. And the third and the fourth, that is Clostridium septicum and Xanthomonas citri, are two different species of bacteria which are again not utilized in genetic engineering for plants. So this won't be the answer. So our final answer for this question will be option number 2, that is Agrobacterium tumefaciens. So Agrobacterium tumefaciens वो बैक्टीरियल स्पीशीज है जो जेनेटिक इंजीनियरिंग ऑफ प्लांट्स में सबसे ज्यादा यूज किया जाता है. होप यू अंडरस्टूड दिस वेल. बेस्ट ऑफ लक. थैंक यू.